Hello everyone, Ramya, Assistant Professor of CSC Department from Rohini College of Engineering and Technology, subject code OMD 551, Unit 5, topic Blood Cell Counters Auto Analyzer. First, objective to learn about the concepts of blood cell counters and their methods of cell counting. Blood cell counters. Changes in the normal functionality of an organism are often accompanied by changes in the body cell count. Here, the blood constitutes 5 to 10 percentage of the total body weight, and an average adult it amounts to 5 to 6 liters. Here also, the blood consists of corpuscles suspended in a fluid called plasma in the proportion of 45 parts of corpuscles to 55 parts of plasma. Then the blood cells are divided into different groups according to their form and functions are shown in the following table. Next, we are going to see the red blood cells. It is in the form of a biconcave disc with a mean diameter of about 7.5 mu and a thickness of about 1.7 mu. Next one is white blood cell. It is a spherical cells having a nucleus. There are normally 500 to 10,000 white cells per cubic mm of blood, but their number varies during the day. The next one is platelets. The platelets are usually tiny, round, oblong, or irregularly shaped cells of the blood with an average diameter of approximately 2 mu. Next, we are going to see some calculation. First one is mean cell value. It is calculated from the PCV and the number of red cells present per liter of blood. For example, just consider if PC is 0.45, that is 1 liter of blood contains 0.545 liters of red cells. And if there are 5 into 10 power 12 red cells are present here, so the formula is applied in the mean volume. So we will get the normal mean red cell volume as 86 plus or minus 10 femtometers divided by 1. The next one is mean cell hemoglobin. It is calculated from the HB and the red cell count. Here also we have one example. They are given as 15 gram of HB per deciliters of blood, and there will be a 150 gram HB per liter of blood, and the number of red blood cells is 5 into 10 power 12 per liter. So MCH equal to 150 divided by 5 into 10 power 12 equal to 30 picogram. Here the normal cell hemoglobin is 29.5 plus or minus 2.5 pg. And the next one is mean cell hemoglobin concentration. It can be calculated if PCV and HP per DL are done. Means for example, here also your PCV is considered for 0.45 and the HB is 15 pg per del. Then that value is applied in the MCHC, then you will get 33.3 gram per DL. Then the next one is mean plate volume. It is the ratio of the integrated platelet volume to the platelet count and is expressed in femtometers. Then we are going to see the next one is platelets rate. It is the percentage of the total specimen volume occupied by the platelets. Next one is red cell distribution width. That means it is a numerical expression of the width of the size distribution of the red cell. Next one is the platelet distribution width. It is related to the size range covered by those plates lying between the 16th and 84th percentile. Then we are going to see different types of methods of cell counting. First one is the microscopic method. It is the most common and routinely applied method of counting blood cells. Even today, particularly in small laboratories, in the microscopic method in which the diluted sample is visually examined and the cell counted. It is also commonly known as the counting chamber technique. It suffers from several common drawbacks. Then the next method is automatic optical method. It is based on collecting scattered light from the blood cell and also convert it into electrical pulses for counting. Here also a sample of dilute blood is taken in a glass container. That means it is drawn through a counting chamber in which the blood stream is reduced in cross section by a concentric high velocity liquid sheath. Here also it provides a dark field illuminated zone on the stream, and the light scattered in the forward direction it is collected to the cathode of a photomultiplier tube. And the next one is Coulter counters. Here we have a wide range of particle counting instruments. It is based on the Coulter principle. Here these instruments range from the small counters used primarily for red and white cell counters in very small hospitals and clinic to the multi-parameter microprocessor. Then the next one is laser based cell counting. It is used to determine the number of RBC, WBC, and platelets. Here, the output of this state goes to the discriminator. Here, the principle used in the laser based blood cell counting is the angle of scatter light. It is different from different side blood cells. Also, it contains the hemoglobin concentration in the RBC, also can be measured by this method. And the next one is auto analyzer. Using this, you can able to measure blood chemistry and display that on a graphical recorder. In the figure, it shows the auto analyzer. First, we are going to see the sampler. Using the sampler, you can able to feed it to the analyzer using the sampler. Up to 128 samples can be placed in the sampler. Then it contains holes in which sample tubes are placed. Then the next one is mixing or proportioning pump. That means it is the heart of the auto analyzer. It also simultaneously proportional to the working on a sample. Here it mixed with the reagents to give proper chemical color reaction. It can be read by a colorimeter. Then the next one is dialyzer. It separates interfacing substance from the sample by permitting the sample through a semi-permeable membrane. And the next one is heating bath. It is used to heat the fluids continuously exactly at 37 degrees Celsius temperature. It is very important because color development depends temperature also. Then the colorimeter. It is used to monitor the changes in optical density of the fluid. It measures color density proportional to substance concentration. Then the graphic recorder. It is used to convert the electrical signal into a graphic display. Thank you.